So thank you for the opportunity to present my research today. As Osama said, my current lab is working on investigating the idea of neurocardiology and understanding neuronal regulation of the heart. We're not doing a lot of specific ion channel work at the moment in regards to that project. So we'll get to that part at the end, but I'm going to start off with some of the work that I did during my postdoc, focused on voltage-gated sodium channels so that we can talk about ion channels today. Let's see. Okay. So in our lab, we're really interested in subcellular localization of proteins. How do neurons get the proteins that they need to build excitable membranes to communicate with other neurons to the exact location they're supposed to be? So neurons have this immense architecture that's a challenge to overcome, where proteins are made in the cell body and then have to get transported along this axon to where they need to be in order for neuronal communication. And voltage-gated sodium channels are a really great example of that. So the picture over here is a hippocampal neuron, and this yellow over here demonstrates the localization of voltage-gated sodium channels. And the red is a surface tag, so you can see that there's a few voltage-gated sodium channels in the somatic membrane, but the majority of the ion channels that you see are in this really high concentrated area of the axon initial segment. And this is necessary for the initiation of action potentials from this region. So my graduate work was investigating how do neurons get this high density of voltage-gated ion channels to this very specific subcellular region of the neuron. And then during my postdoctoral work, I moved over to peripheral sensory neurons to ask, how do neurons regulate the location of voltage-gated ion channels when you have these really incredibly long axons? So the cell bodies are sitting near to the spinal cord, and then they have to extend these long distances down your arms or down your legs. And the site of action potential initiation is actually long distances away from that cell body. So just a reminder of the architecture of neurons: you've got your cell body, you've got dendrites around it, and you've got your axons. And in the central nervous system, again, the information is flowing from the cell body away down the axon where it synapses on its target, which could be another neuron. In the peripheral nervous system, we're talking about neurons with this pseudo-unipolar morphology, where you've got these sensory receptors at the distal axon. This is where the action potential is actually being initiated, and then the information is flowing back towards the soma, past the soma, to the central connections within the spinal cord. So like I talked about, I'm first going to talk about voltage-gated sodium channels and sensory neurons. And then towards the end, I'll talk about the project over here in the sympathetic nervous system. So just to give you a sense of the architecture of the nervous system, just a reminder, you've got the central nervous system, you've got the peripheral nervous system, and then today we're going to be looking at two distinct branches of it. The first part of the talk will be about the sensory system, and then the second part of the talk will be about the sympathetic system. So to start off, the first half is about neuroplasticity of sensory neurons, and like I alluded to, altered trafficking of voltage-gated sodium channels in the context of neuropathic pain. So the first part of this will be neuropathic pain. And then I'll talk a little bit about a project we had looking at chemotherapeutics and their role in chemotherapy-induced neuropathies. So why NAV 1.7? So the Waxman lab, where I did my first postdoc, was really focused on several isoforms of the voltage-gated sodium channels, but specific interest in NAV 1.7 in the sense that it can be a volume knob for pain. So this is a Wired article where they interviewed two human patients, or two people, one with a mutation in their voltage-gated sodium channel NaV1.7 that caused painful neuropathy, where putting their hand under cold water could cause extreme pain, or sorry, under warm water could cause extreme pain. And then another individual with a mutation that caused a loss of function mutation, where they had an insensitivity to pain. And so basically functional mutations in this gene can regulate the amount of pain that an individual feels. So why NaV1.7? So this is showing an action potential and how the different isoforms of NaV1.7 contribute to it. And NaV1.7 in particular amplifies the subthreshold depolarization. So it's involved in kind of gating when action potentials are generated. And then another sodium channel isoform that's highly implicated in neuropathic pain is NaV1.8, and it contributes to this depolarizing phase of the action potential. So together, these sodium channels kind of regulate the excitability and rising phase of the action potential in peripheral sensory neurons. And like I talked about before, loss of function mutations can cause congenital insensitivity to pain, and gain of function mutations can cause disorders such as inherited erythromyalgia, which is called man on fire syndrome, or extreme pain disorders. So again, we have this idea that the activity level of NaV1.7 really fine tunes the ability for individuals to feel pain or not. So in terms of the dorsal root ganglia, where would we find NaV1.7? Immunohistology shows that it's at these sensory endings far from the soma. You can actually see it within the cell body as well. And then if you look at the central projections, it's at these terminals within the spinal cord. And so the question we're really interested in is, how does NaV1.7 get distributed along here? And particularly using antibodies to look at distribution, you can see there's a lot of it intracellular in the cell body. And so we really needed tools where we could specifically look at the surface distribution of the sodium channels, which are the subset of the channels which would actually be functionally contributing to ion channel excitability. So I'll be talking to you today about a series of tools and techniques that I initiated during my postdoc in the Waxman lab, and then a couple of very talented MD, PhD graduate students in the lab really took this technique and expanded on it. I worked mostly on enterograde trafficking, and then they added a lot of different variations to this technique to be able to look at retrograde trafficking, look at rates of insertion and protein removal, and a lot of different aspects in terms of the life cycle of ion channels. So like I talked about, we really needed tools where we could look at the subset of voltage-gated ion channels on the neuronal membrane. So one type was had a little extracellular loop with a biotin acceptor domain. So if you co-transfect with a biotin ligase, it will biotinylate the lysine residue in the center of the sequence, and then we can use streptavidin conjugated fluorophores to specifically label channels on the cell surface. And then another technique we came up with was tagging the NaV1.7 with a halo tag, and we put the beta 4 accessory subunit here, we tethered that to the alpha subunit so that we could get this halo tag extracellularly and be able to use cell impermeable ligands to specifically label surface proteins. So the halo tag here, it's about approximately the size of a GFP, and it's an enzyme where when you add the halo tag ligand, it will react with the functional group on this halo tag ligand and create a fluorescent, a covalent bond. And we use a flavor of it. You can have a lot of different tags that are associated with it, but we specifically use these fluorescent ligands. And importantly, we have both cell permeable and cell impermeable versions. So it gives us a lot of flexibility in being able to tag subsets of these ion channels and ask where they are and where they're going. So the first thing we did, and this is one of the few electrophysiology slides you'll see during this talk. I apologize, we don't do a lot of electrophysiology. We first wanted to make sure that the addition of these tags didn't significantly alter the function of these proteins. We get slight variations in the current density of these channels, but the other biophysical characteristics of the channel are not changed with the addition of the tags. And then this is an example of how we can use the cell permeable versus impermeable tags. So if we first put on a cell impermeable tag, we can specifically label the surface population of NAV 1.7. And then if we add a cell permeable one, we can see the intracellular pool, and merged, we can see the total amount of NAV 1.7 protein in our neuron. So in order to answer the question of how do neurons get voltage-gated sodium channels to their distal axons, we came up with this novel trafficking paradigm that we called opal imaging, or optical pulse-chased axonal long-distance imaging. So this really relies on both the use of that halo-tagged sodium channel that I talked about, combined with the growth of dorsal root ganglion neurons in this microfluidic chamber. So the cell bodies are plated over here in the somatic chamber, and then their axons grow through these microbarriers in this chamber into the axonal part of the culture. And then we label with a cell permeable ligand, specifically in the somatic chamber. And so that's going to be able to label any proteins that are there, whether they're on the surface or intracellularly. And since we're interested in vesicular trafficking in this experiment, we do our imaging over here in the axonal chamber after we give some time for channels that are in these trafficking vesicles to move from the somatic chamber to the axonal chamber. And so the only signal that we see over here in this axonal chamber is vesicles that are actively transporting during our experiment, moving interogradely along the axon. So when we do our imaging, it looks like this. So each of these dots is an individual trafficking vesicle containing a fluorescently tagged NAV 1.7. So you can see it works very nicely to see vesicular transport along the axon. And then we quantify the movement using these chimegraphs, or these time over distance plots. And so just to orient you, anything that's not moving during this time course, so we have one vesicle that's not really moving here, that's going to show up as a straight line because it's not moving in space during the time course. And then as vesicles are moving all across the axon, we can see that as these diagonal lines. And so the angle of these lines, or the slope of these lines, represents the velocity that these vesicles are moving through along the axon. So this is looking at the end of an axon, as they move along, they kind of come to the end and start to aggregate here at the end. And then we can see some that turn around and head back the other way after they've spent some time here. So in this chymograph, you can see some of the slopes going the other way suggest retrograde trafficking, suggesting that we get this kind of trafficking loop, if you will, or we have this kind of roaming population of vesicles that haven't delivered their proteins to the cell surface. And then we wanted to investigate. It seemed, based on the speed of these vesicles, that they're probably being moved along the axon using kinesin-based transport on microtubules. We wanted to confirm that. So we took axons and incubated them with either DMSO or the microtubule depolymerizing drug nicotazole. And in the same axon, after 60 minutes of incubation, we still saw trafficking along the axon in neurons incubated with DMSO. But any incubated with nicotazole, almost all of the trafficking stopped. And I like this chymograph because we still have some vesicular movement, meaning that the neuron is still alive, they're still able to be transport, but the majority of microtubules have been depolymerized. So the next thing we wanted to ask was what kind of transport vesicle was carrying NAV 1.7 along the axon. So we turned to Rab proteins. So these are small GTPases that help to regulate membrane trafficking. And there's different Rab proteins that are associated with different parts of the cell cycle. For example, Rab1 and Rab2 tend to be focused on ER to Golgi transport. And then there's several that are implicated in anterograde trafficking or retrograde trafficking, or recycling endosomes and such. And so what we did was screen several of the most promising Rabs to determine what subtype of vesicles these sodium channels are being transported in. So we co-transfected this halo tag sodium channel along with EGFP tagged Rab proteins. And in the screen over here, we looked at several different proteins, and we found that Rab6 most often co-transported with the sodium channels. So in the chymograph, you can see that their tracks are basically overlapping, demonstrating co-transport. And then this is an example of EGFP Rab3A. And if you look at the tracks in the chymograph, they're distinct from each other, demonstrating that they don't show co-transport. So we're able to identify that NAV 1.7 is most often trafficked in Rab6A positive vesicles. So then we were interested in whether there's dynamic regulation of this. So if you think about, you get a cut on your finger, it gets a little bit inflamed, and you have increased sensitivity there. Is NAV 1.7 involved in this? And other studies using immunohistochemistry or Western blots had shown an upregulation of NAV 1.7 in the context of inflammatory pain. So we wondered if we could see this process in real time and understand what's happening to underlie this upregulation of NAV 1.7. So here's your other electrophysiology for the talk. So in control cells, we got a certain amount of NAV 1.7 current, but after only 4 hours of incubation in cultured cells, we get this huge upregulation or potentiation in the current for NAV 1.7 in response to this inflammatory cocktail. So we wanted to ask, can we see this process happening in real time? And indeed we could. So doing this opal imaging on axons and control cells, we do see obviously some trafficking, but when we incubate for only 4 hours, we can see this huge potentiation in the amount of trafficking vesicles there. So these chymographs demonstrate the movement of vesicles along these axons during these image sequences. And then here in panel B, we're showing that a lot of this protein trafficking is accumulating here at these axonal endings, showing that a lot of these trafficking vesicles are making it all the way to the end and accumulating at the end. And again, this is in a sensory neuron where action potentials would be initiated physiologically. So we quantified that axonal trafficking, and we saw an increase in the flux, which is the number of vesicles moving along the axon per unit time. We saw an increase in the velocity in neurons that had been exposed to inflammatory mediators. And we saw an increase in the fluorescence intensity for these vesicles. And E is showing that we put the halotag ligand just on glass and then photo bleached them, and then looked at the fluorescence intensity of the bleach steps to figure out what the intensity is for a single molecule. And then we extrapolated that to the fluorescence intensity of our vesicles. And this suggested that most of the vesicles that we're imaging under control conditions have about 1 channel or fluorescently tagged channel. And then with the addition of inflammatory mediators, this was increased to about 3 channels. So again, we're seeing this huge potentiation in trafficking, which means more vesicles, they travel faster, and that there's more channels per vesicle. So I demonstrated that these vesicles accumulate at the end of the axon, but that doesn't translate into increased excitability unless they're inserted into the axonal membrane. So we wanted to ask, do we see more surface channels in response to inflammatory mediators too? So we used the surface labeling strategy again, and when we quantitated the fluorescence intensity, we saw both total protein as indicated by the venous or the GFP, and an increase in the amount of surface protein as indicated by labeling with a surface-specific label. So a follow-up project looked at how does this increased accumulation happen? Can we see an increase in the amount of insertion? And is there a correlated increase in the amount of internalization also? So the proteins that get put on the surface, are they staying there longer? Or is there an equal amount of internalization? So this is an assay that was developed by Grant Heigard. And so what happens is that you put in the axonal chamber, we're putting in a cell impermeable ligand that's going to label any of the proteins that are on the cell surface. And then this is washed off, and a second label of a different color is added to the bath. But all of the binding sites of the proteins on the surface are already taken, so those can't take up the second signal. But any new proteins that are inserted in the membrane during the time of imaging can take up this new signal. And so if we do time-lapse imaging, we can see an increase in this label 2 that will demonstrate the amount of protein insertion at this axonal ending. And then the decrease in the fluorescence intensity of label 1 will show how much of this protein has been internalized during that same time. So this is what it looks like. On the top, we're looking at the label demonstrating insertion of protein. So at the beginning, we don't have any protein or very little protein that we can see on the cell surface using the second label. And then over the course of 6 hours, we can see that signal getting brighter and brighter. And if we look at control axons relative to inflammatory mediators, we can see that starting about at this 4-hour mark, we're getting this increase in the rate of internalization of proteins, which is corresponding to that increase in trafficking that I was showing you before. And if we look at the proteins already on the cell surface, we can see the rate of internalization. And this rate doesn't seem to change with the presence or absence of the inflammatory mediators, suggesting that this increase in the density of ion channels at the distal axon is due to increased protein insertion and not due to changes in internalization. And again, we were interested in whether this correlates with neuronal excitability. So this is another experiment done by Grant, and he put inflammatory mediators on for 4 hours, then he washed it off. And then he looked at spontaneous activity in these distal axons as reported by an axon-specific GCAMP indicator. And so the time series here is showing, you can see an increase and decrease in the calcium signals, suggesting potentially an initiation of an action potential in this specific axon. And then after looking at spontaneous activity, he added on the Protox2, which is a specific inhibitor for NAV 1.7, and then again looked at spontaneous activity. So what he found, what these are indicating is each of the lines on here is showing the activity for an individual axon over the imaging frame. And so under control conditions, we get some spontaneous activity, but if you look over here in axons with inflammatory mediators, we have a lot more axon endings that are active with a higher frequency of events. And that's indicated here that the control axons showed little spontaneous activity, and this was potentiated with incubation with inflammatory mediators. And both of these were inhibited by the NAV 1.7 specific inhibitor, suggesting that this enhanced excitability is being driven by this increase in the amount of NAV 1.7 being inserted on the cell membrane. So that's going to end the first part, looking at just overall trafficking of NAV 1.7 in sensory neurons, and demonstrating, so I demonstrated that NAV 1.7 channels, we can see their trafficking along the axon. They're being carried in RAB6 containing vesicles, and that even a short incubation in inflammatory mediators can increase the amount of protein trafficking, insertion in the membrane, and excitability. So any questions on that part before I go on?
We are starting to have questions, but we'll keep them till the end if you don't mind.
Okay, perfect.
Yeah.
Okay, so the next part is going to be looking at chemotherapeutics. So we wanted to ask, could we see the molecular basis for chemotherapy-induced neuropathies? So a lot of patients that undergo paclitaxel treatment, this chemotherapy drug, experience chemotherapy-induced neuropathy to the extent that many patients stop taking the treatment, or it reduces their ability to tolerate it and reduces their quality of life. So paclitaxel works by stabilizing microtubules, which in cancer cells prevents their division, but this causes the off-target effect on cell types such as neurons. And then other groups have demonstrated that incubation of neurons, rats that have been exposed to paclitaxel, if they look at the dorsal root ganglion neurons, they can see an increase in the amount of NaV1.7 protein in their dorsal root ganglion neurons. And then looking within the spinal cord, you can see increased amounts of voltage-gated sodium channel expression within the spinal cord. So again, we wanted to ask, can we see this process happening in real time? So the first thing we needed to do was look at what a good amount of paclitaxel or a good concentration of paclitaxel would be. In patients, concentrations can reach up to 4 micromolar, but the steady state plasma concentrations can be in 2 to 40 hours after treatment, or somewhere between 20 and 800 nanomolars, according to this publication. And again, this might be in the bloodstream. And so how much of that is actually getting to the neurons? So we don't really have an answer to that, but we tried 2 different concentrations, a lower concentration and a higher concentration. The first thing we did was just look at neuronal health. And in just DMSO, we didn't see a lot of axonal degradation. With a low concentration of paclitaxel, we saw a little bit of axonal degradation, but we saw much more degradation, especially in the later time points with this higher amounts of paclitaxel. And there is a known amount of retraction or degeneration of axons in human patients after paclitaxel treatments. So then we wanted to know what these different concentrations, how they affected the expression of NaV1.7 at these distal axons. So again, we used the cell impermeable halotag ligand and looked at just protein levels in these distal axons after incubating with paclitaxel. So with the 25 nanomolar paclitaxel, we saw a fairly large increase in the amount of sodium channel proteins at these distal axons, both at 24 and 48 hours. We saw some increase, not significantly, for the higher concentration of paclitaxel, and then we saw lower concentrations at the 48 hours, which corresponds with this axonal degradation we saw with this higher concentration. So then we looked at protein trafficking to see if that would correspond with the amounts of surface protein that we saw. And we saw increased in the number and the speed of trafficking as early on as 6 hours. And then we saw the most dramatic increase in the amount of protein trafficking for the lower concentration of paclitaxel. And this, we largely didn't see this with the higher concentration of paclitaxel. So we saw fewer vesicles moving along the axon in these with the higher concentration of paclitaxel, which correlates with the axonal degradation. So probably if these axons are degrading, you're not getting a lot of protein transport. In terms of speed, we saw consistently higher speeds relative to controls for the lower concentrations of paclitaxel. And then even at 48 hours, we saw very few vesicles moving along the axons, but the ones we saw were moving quite fast, suggesting that any of the remaining microtubules that were still there were still able to transport sodium channels to the distal axon. So Inflammation is another part of chemotherapy treatments. So we wondered, since we saw an increase in trafficking with chemotherapy, and we also saw it with inflammatory mediators, are these additive to each other or not? So under control conditions, again, we see this potentiation in the amount of fluorescent protein at these distal axons, just in response to inflammatory mediators. And this, we saw a potentiation above and beyond what we saw for the paclitaxel, both at the low concentrations and at the high concentrations. And we also saw a similar trend when we looked at trafficking. So if we looked at the number of vesicles that were moving along the axon, again, inflammatory mediators will potentiate the trafficking, and this is even further potentiated above and beyond what we saw for the paclitaxel treatment, both for the low and high concentrations. And we saw that trend both for the number of vesicles and the velocity of the vesicles, demonstrating that the presence of inflammation at the same time as chemotherapy treatments could negatively affect the development of chemotherapy-induced neuropathy. So running a little short on time, so I'm going to jump through the conclusions. And then next I'm going to talk about the project that I've built my current lab on. So this is changing gears a little bit, and we're moving into the sympathetic nervous system. And instead of looking at ion channels, we're looking at the trafficking and synaptic localization of neuropeptide Y. So during cardiovascular disease, so say for example, there's a myocardial injury that's picked up by your sensory neurons and communicated through the central nervous system to your autonomic nervous system. And this can cause long-term remodeling of your autonomic nervous system, including structural changes to your sympathetic nervous system, including cell size, an increase in cell size, and an increase in excitability. And the opposite is true for the parasympathetic nervous system, where we get this decrease in excitability. This can show up as altered neuropeptide or neurotransmitter production, and this can have a negative feedback loop to the heart, where you're getting excess excitable signals to the heart, which can cause further injury and potentiate this cycle. So a quick overview of the sympathetic nervous system. You've got central inputs. Those come down through your preganglionic neurons into your spinal cord. And these preganglionic neurons synapse onto your postganglionic neurons that are sitting in this ganglia outside of the spinal cord. And then they project long axons to their target organs, such as the heart, which we're interested in. And if you look at the junction between these axonal endings and cardiomyocytes, specifically in the sympathetic part, we've got release of norepinephrine or noradrenaline onto beta-adrenergic receptors on the cardiomyocytes. And along with it, we get this release of neuropeptide Y onto Y receptors on the cardiomyocytes. And this is what we've really focused on in our lab, is can we look at the trafficking of NPY along these axons, and can we look at their accumulation at the varicosities at these neurocardiac junctions? So why do we care about NPY? Several studies have demonstrated that elevated levels of NPY in the coronary arteries, suggesting that it's been released from sympathetic neurons, and this is the spillover into the bloodstream there. So increased amounts of neuropeptide Y is correlated with negative outcomes in human patients in heart failure, myocardial infarction, and other diseases, suggesting that increased sympathetic tone is regulated with or correlated with negative outcomes in human patients. And neuropeptide Y is thought to be proarrhythmic, so it can contribute to sudden cardiac death. It's present in dense core vesicles, and under normal conditions, sympathetic neurons tend to release norepinephrine or noradrenaline onto the beta-adrenergic receptors. But under conditions of higher frequency stimulation, you also get release of neuropeptide Y. And this paper is suggesting that maybe in order to prevent arrhythmias after something like a heart attack, maybe instead of just using beta blockers, we should also be blocking neuropeptide Y. All right, so back to the theme that we were talking about before is asking how these proteins get where they're going. And in the case of sympathoexcitation, if we're releasing increased amounts of neuropeptide Y onto cardiomyocytes, this suggests that we probably have increased trafficking of neuropeptide Y, and we have the creation of more neuropeptide Y in the cell body. So we want to know, can we see this process happening in real time in a similar way that we saw for the NAV 1.7 trafficking vesicles? So the model we're using in our current lab is the spontaneously hypertensive rat model. So this is a neurogenic model of hypertension. These rats, as the name suggests, spontaneously develop hypertension early in life. And if you block the activity of the sympathetic nerves, it actually blocks that development of hypertension. So this is really cardiovascular disease that's driven by an overactive sympathetic nervous system. And then we're comparing everything to the control strain, which is the Wistar-Kyoto rats. And this is a demonstration that these sympathetic neurons maintain their hyperexcitability even in culture. So even cultured neurons from neonatal animals, when challenged with elevated extracellular potassium, show increased calcium responses, suggesting increased excitability. And importantly, early animals, pre-hypertensive animals, show increased release of both norepinephrine and neuropeptide Y in their coronary arteries, suggesting that their cardiac sympathetic neurons are overactive in this model. So we're going to culture and transfect in these microfluidic chambers, as I just showed you. And then we're going to perform similar opal imaging techniques, this time looking at trafficking vesicles that have a halo tag neuropeptide Y contained in them. So again, we can see nice axonal trafficking of neuropeptide Y containing vesicles moving along the axon. And then the question is, can we see a difference between the SHR animals and the normotensive animals? So this is the control strain, the WKY strain, showing protein trafficking along the axon. And then hopefully you can appreciate with the spontaneously hypertensive rat model, we get this huge potentiation in the amount of trafficking of NPY containing vesicles. And I'll show you the quantitation on the next page, but you might be able to appreciate that there's more vesicles, they're moving faster, but they're not quite as bright, suggesting we actually have fewer proteins per trafficking vesicle, which is the opposite of what we saw for the NAV 1.7 under an excitatory condition. So again, this is quantitating the velocity, showing the enhanced speed of the vesicles, the enhanced flux, but the decrease in the fluorescence intensity of these vesicles. And again, we wanted to demonstrate that the trafficking we're seeing is kinesin based. So like I showed you for the sodium channel, if we put on nicotazole, we can basically stop all trafficking in about 60 minutes relative to the control, DMSO control, where we can still see trafficking maintained. And then just a quick taste into where we're going with this. We're working on developing these co-cultures. So we've got sympathetic neurons here growing across, and we've added cultured neonatal cardiomyocytes to the axonal side, where we can look at neurocardiac communication. So this is looking at calcium imaging on that axonal side. These lines here are axons that have taken up this calcium indicator dye. And then these are neonatal cardiomyocytes that beat spontaneously in culture. And then if I zoom in to part of it, you can actually see the axon here light up prior to a calcium event in the cardiomyocyte, suggesting we do have these functional neurocardiac junctions that we can investigate. We also wanted to look at, can we modulate the system? So the two sides of this microfluidic chamber are fluidically isolated. So anything we add to the somatic side, the axonal endings and the cardiomyocytes won't see. So we can add nicotine to activate our nicotinic acetylcholine receptors on our postganglionic neurons here. So in this movie, what you're seeing is spontaneous activity of the cardiomyocytes up here with some axons growing over them. And at about 20 seconds is when we add nicotine to the somatic chamber to activate the neurons. And hopefully you can appreciate this increase in cardiomyocyte activity in response to the stimulation of our sympathetic neurons. So this just demonstrates that we do have a system where we have functional neurocardiac junctions. And the next question we're going to get to is, how does that affect neuronal trafficking? If we stimulate these neurons and stimulate them to release their neuropeptide Y, how long does it take for them to refill these synaptic vesicles? And what are the differences in trafficking in the condition of sympathoexcitation versus normal axons? So we need to figure out a way that we can label the cardiomyocytes better. Right now you can just see some autofluorescence in the cardiomyocyte, and hopefully you can see it spontaneously beating here. And then what you're seeing here is the opal imaging of neuropeptide Y containing vesicles that are moving along, and these enlargements here represent synaptic varicosities sitting on top of this cardiomyocyte that have accumulated neuropeptide Y vesicles. So we can start to ask questions of, how do these vesicles know which synaptic varicosity to get captured in? How can we release them? How long does it take to refill these, and other such questions? All right. So I know that was a few different topics, but the thread weaving it all together is the fact that neurons that are hyperexcitable show increased amounts of trafficking, whether it be sodium channels or neuropeptide Y. And we've developed these tools where we can be able to look at the changes in real-time neuroplasticity at a molecular level in both sensory and sympathetic neurons. And you know, the overarching goal is, can we identify what are the molecules that are involved in this trafficking, and is there anything we can do to modulate the trafficking in the case of voltage-gated sodium channels? Can we inhibit the trafficking of pain-related sodium channels as a modulator for neuropathic pain? And in the case of the sympathetic neurons, can we modulate neuropathic or NPY trafficking so that we can reduce sympathoexcitation in the context of cardiovascular disease? All right, and so huge thank you to my lab and all the work they're doing. And thank you for the NIH Heart, Lung, Blood. I'm currently funded by a CATS award. We have a predoctoral fellowship to my grad student Nancy from AHA. And thank you for your attention.
Thanks, Liz. That's a beautiful talk. Thanks so much. We have a number of questions, so we'll since we only have a few minutes, I'll try to go through them relatively quickly. So the first question is from Rohit Kumar. Is action potential propagation in peripheral versus hippocampus different from a kinetic standpoint?
It's a really great question. And I don't think I know the answer. I'm sorry.
No worries. Second question from Mark Rogers. Are the axonal endings in your DRG sensory neuromicrofluidic chamber cultures more typical of peripheral, like the skin, or central, like the spinal cord and nerve terminals?
That's a great question too. We always thought of them more as peripheral. We played around with trying to do like a triple culture where we could put some keratinocytes on one side and we could put some motor neurons or something from the central spinal cord to try and potentially, get this differential distribution for morphology of these neurons, but we didn't get too far with that. So yeah, it's an incomplete system, and we just tend to think of these free endings as more peripheral.
Okay, more like the skin, as you said.
Yeah.
Okay. Another question is also from Rohit Kumar. Also, how do they fit in context to your hippocampal versus peripheral neuron comparison? What I'm trying to ask is whether where is the molecular machinery located to produce the required molecules, proteins, for example, and how or what is the sequence of such events if it was centriole controlled?
Yeah, the understanding is that in both of these cell types, the synthesis happens in the cell body. In the ER, goes to the Golgi, gets packaged into the vesicles, and gets sent off. Obviously, the peripheral sodium channels have a lot further to go, and I didn't get far enough in the project during my graduate studies to know which motor proteins are involved with trafficking to something like the axon initial segment, and whether that's similar or different to the pathways that are taken in the peripheral neurons. And that's something that certainly should be investigated at some point.
Yeah. Another question from Hein Nguyen. Thank you for a great talk. Do you know if any of the gain of function or loss of function mutations in NAV 1.7 affect trafficking?
I can't think of one. Oh, yeah. We did look at one that it's actually an interesting temperature sensitive mutant. And we found that the mutation both alters the activity of the channel and alters the trafficking of it. We don't know how or why, but yeah, there seems to be some mutations that can alter trafficking.
Another question from anonymous attendee. You mentioned that NAV 1.7 channels accumulate at the distal axonal end. I was wondering what their role is there. I think you mentioned increased spontaneous activity via calcium signaling, but can you say anything about their effect on firing frequency?
The experiment I showed demonstrated increased spontaneous activity at these free endings, which I guess could be considered, an increase in firing frequency. But whether that's, going to be maintained in vivo or something like that, we haven't figured that out yet. So.
Sure. Another question from Rohit Kumar as well. Is it possible that the morphology of the DRG neurons can facilitate higher fluxes?
The morphology can facilitate their flux.
Higher trafficking. I'm assuming trafficking.
I don't think I quite understand.
I'm not sure
The morphology. So just in general, whether they traffic more, my observation so far in working with both sensory and sympathetic neurons is that sensory neurons are built to traffic, right? Like we just see trafficking vesicles just go. We have to kind of stimulate sympathetic neurons and play some tricks to really be able to see trafficking in sympathetic neurons. So it suggests there is this kind of inherent difference in how these neurons process and set up their excitable membranes, if you will. I don't know if that was the question, but
Yeah, sure. Guy Salama is saying, beautiful talk. You test inflammatory biomarkers using the cocktail that you mentioned. Did you test these kind of components separately? And if so, does this affect in a different way the trafficking?
Yeah, it was always on my list of things to do, kind of tease apart what was there. I ran out of time during that project. Follow-up projects have looked at, potentiation with like TNF alpha and some other compounds. Another thing I wanted to do was, what if you put the inflammatory mediators only on the soma or only on the axon? All the data I showed had it in both compartments. So certainly a lot more that we can do with it and ask a lot of more questions about the specificity of the response.
Sure. Another question by anonymous attendee. There seem to be a large population of vesicles that do not move along the axon. Do you know what is controlling the vesicles to stop in the axon during trafficking? Are these localizing to the nodes?
So we were looking at just all DRG neurons. The small sensory neurons tend to survive better in our culture conditions, which would be the small non-myelinated neurons. So we wouldn't anticipate that they would even physiologically develop nodes. And we aren't looking at, we haven't included co-cultures to myelinate them. So these are unmyelinated cultures. So I don't think they'd be stopping at what we would consider nodes. But really the reason we have to do this opal imaging is because there is just this kind of large intracellular pool of vesicles that are there, whether it's in the soma or along the axon. And you can't really see this trafficking if you let too many of them build up. And we don't really know. It could just be, you want a good number of them there when you need them. There could be kind of this recycling pool that's always available. And but yeah, I don't know why do they stop? Why do they stop where they do? It's also a really good question.
Sure. Last couple of questions. One from Pablo Gaiti. Great talk. Since MPY can co-localize with noradrenaline in the same vesicle, I wonder if, in addition to affecting trafficking properties, the ratio of neuropeptides release is also changed co-transmitter.
Sorry, what's the question again?
So since MPY can co-localize with noradrenaline, NA, I'm assuming, in the same vesicle, I wonder if, in addition, by affecting trafficking properties, the ratio of neuropeptides release is also changed yeah.
So the ratio between NPY and noradrenaline, maybe?
That's my understanding. It could be the cardiac outcome, could be the cardiac outcome reflect a synergistic effect of noradrenaline and neuropeptide more than
absolutely.
what neuropeptide Y.
Yeah, so if we're getting increased trafficking of neuropeptide Y, yeah, I guess maybe, does the decrease in the amount of neuropeptide Y per vesicle suggest maybe an increase in the noradrenaline load or something like that? That's a good thought. What we're trying to set up is being able to use amperometry to be able to look at concentrations of release of noradrenaline and neuropeptide Y in real time. So check back in a couple of years and I'll have a better answer.
Sounds good. The last question is from Sergei Palchevsky. Thank you for a nice talk. Can you speculate a bit more about particular structural reasons for NAV 1.7 with RAB6A selective interaction?
Structural reasons. I don't know. See, I think we did a follow-up study trying to look at whether RAB6A is required for trafficking using constitutively active and dominant negative versions of RAB6A, and that didn't really go anywhere. So it just seems like RAB6A is, kind of along for the ride as far as we can tell. I can't speak any more to that.
Sure. That's all the questions we have. So one more time, we are a few minutes after the hour, but we would like to thank you one more time for really a great talk, and as shown by the many questions that we received. So thank you very much, Liz, for coming and accepting the invite. Thank you.
Thank you for the opportunity. Appreciate it.
Of course. Thank you.